minutes call. Start.
Good morning. Today we'll see a topic in cell structure and function under the name biomolecules. Okay. Now these biomolecules, the topic is biomolecules. Now, as you know, this is partly your chemistry and biology concern, how these chemical substances are involved in the living system. Okay, that is your biomolecules. Now, this biomolecule chapter, as you know, first of all, what they did at the beginning, they took the living tissue and the non-living, comparison of non-living and living matter, chemicals in the non-living world and the living world. So when you compare the living and non-living, you know very well, they took a sample of sand to analyze the different components present in that. So they took a animal or plant tissue. How will they take animal or plant tissue? You take a plant tissue, burn it up, and you'll get the ash. So when you get the ash, you analyze the different substances present in that. So when you compare a non-living material and a living tissue, they found out commonly elements. Both of them have got the elementary level substance because you are analyzing in the molecular level, still elemental level, atomic and level we are going. So the chemicals they found out carbon, hydrogen, oxygen, nitrogen they found out in both. So first is a comparison between, first as you go, compare between first they compared non-living matter, say a soil and a living tissue. Living tissue can be a plant or an animal. So when they compared it, they found out many elements like carbon, hydrogen, nitrogen, oxygen, and some salts, mineral salts in the form of mineral salts, ions. So both are present in both. And water is maximum. And when you compare this, or you have found some minerals. As you know, the minerals are in the soil. And minerals, it won't be in an ionic form. The minerals are in, let's say, salt forms. We call that as a sodium chloride, bicarbonate salt, carbonate salt. All these are the minerals. Then they found out one thing is abundant, is present always in the living tissue. That they found out too. There is carbon is present in all the living tissue. Present in all living tissue. Living tissue. Second, they found out what they found out in this, water is abundant, plenty in the living world. Abundant in the living world. In the living world. So this is the main generalization when you compare non-living and living matter. Then they wanted to compare living tissues. Take the living tissue and start analyzing it. So when you start analyzing it, now they found out a number of substances. Will they take? They take an animal or a plant tissue and hydroxychloric acid, smash it and make a paste with acid, and then they filter it and take a filtrate, analyze the filtrate. So when they were analyzing, they found out now there are many substances in a molecular state. What do you mean with molecular state? When you write the C, we won't write an atom of C, also C and C4. Though valence is 4, we won't write C4. Yeah, Na only molecular atom. So they found out many of the elements, major elements they found out. So second what they did, analyzing the tissue, analysis of living tissue. Living tissue, same thing in the plant or animal tissue. What will they do? First, they heat it, warm it up. So water in that content will become dry. Water is lost. then the remaining tissue, when they burn further, it will become an ash. So they subject this ash for analysis. Maybe how will they? You have a solvent, they dissolve it, and then they filtrate it. Some may be in the filtrate, some may be remaining on the supernatant. It will remain unfiltered. So they compare the elements in the unfiltered component and the filtered component. Now, so when they analyze, they found out there are many substances or the molecular elemental stage, particularly carbon, hydrogen, nitrogen, oxygen, N2, okay, hydrogen and oxygen, and a number of salts. Salts in the form of sodium chloride, that is potassium chloride, say potassium chloride, KCl, or MgCO, magnesium carbonate. So a number of salt apart from that they found out. That means when they analyze, carbon is present in all the living tissue. So the major element they found out carbon along with these substances, the other mineral salts also are present. So they came to the conclusion it's not only these major substances are essential. You need other than this carbon, hydrogen, nitrogen, salt in NaCl, KCl form. So this is what you will be studying in the nutrition lesson: macronutrients and micronutrients. Macronutrients are the elements of carbon, hydrogen, nitrogen as the macro elements which are needed in a larger quantity. Micro elements are salts or anions like potassium and sodium. Same thing, they came to the analysis. There are certain substances in a larger major elements, apart from this, there are minor salts. Then they wanted to find out what is the difference in this. Some of these substances, they get involved in the biochemical reaction in the living world. Now, when you compare non-living and living world, anyway, before you take the analysis of the living tissue, two things are common in the living tissue. That is metabolism, chemical reaction, and the body constitution. That is what the living tissue is made up of, as well as what are the chemical reactions going on in the living tissue is common to all the living tissue. So they wanted to see, after keeping that in your mind, they started analyzing living tissue. They got all this. Then they wanted to know what is the major difference between these elements. Major difference is some of them get involved into the biochemical reaction in the living tissue. So these substances are getting biochemical. So first comparison of non-living and living came to the conclusion carbon is present in all, though it's present in the non-living world, is present in all the living tissue, and water is abundant in the living world. Then they started analyzing living tissue. How do they take the living tissue? Was heated up, water evaporated, it becomes dehydrated. Then dehydrated salt is burnt and becoming an ash. Either how will they dehydrate? Burn it up. When they burn, water goes up. So you left ash, and that ash is subjected to analysis to find the different element chemicals present in there. How will they? That salt ash could be dissolved in a solvent, the hydroxychloric acid, or particular solvents they can use, different organic solvents, or acid. Mainly acid. Only they use to solve this ash. So when they dissolve it in the acid, and they, after dissolving it, they filter the mixture. When they filter it, some of them with whatever is salt in the acid will be filtered. Whatever is insoluble will remain as a supernatant. So they separated the undissolvable part, which is filtered, as well as the liquid, which is filtrate. So both they separated. They subjected both the things for analysis. Why these things are not filtered? These chemicals are not getting filtered. Why these chemicals are simple thing? What you know? These are soluble and they are not soluble. That's all. So if they are fat or acid insoluble, why are they acid insoluble? What is the importance of it? That's further study. So when they first of all when they found out these different elements, they wanted to find out one major difference. That is, some of these elements are involved in the metabolic pathway. What is meant by metabolism is nothing but chemical reaction. See, some of these elements involved in completely chemical reaction of the body. I'll go through this back again. Now, first comparison is living and non-living. First step, compare living and non-living tissue, non-living matter. Non-living matter. That is the living tissue and non-living matter. What conclusion they came to? The conclusion is carbon is present in all living world. Is in all living tissue. Then water is abundant. In the water is the abundant substance, is the abundant or plenty. Abundant in the living world. In living tissue. Living world as it is. A living tissue. Then second step what they did, the analysis of living tissue. Living tissue. Now how do they analyze? You know very well water is plenty. So you dehydrate water. That is step is what? Dehydrating. How did they analyze? Dehydrate. So remove water. Remove water. Then you get the ash. And ash is dissolved. Dissolved in acid. In acid. Particular acid they can use. And then filter. Filter, and you get, it leads to filtrate, that is a filtrate. Filtrate is the one which is getting down, and supernatant, that is the unfiltered part. Unfiltered part. So they started analyzing the filtered one and unfiltered one. Before, when they found out, they came to the conclusion. Now, when they found out some of these substances, what are they? That micro and macro elements undergo chemical reaction, chemical change in the living body. Chemical change in the living body. The living body. Okay, now they call them as, and when they undergo the chemical change, this chemical change is a definite reaction. That is, a chemical change has a particular direction, it goes in a particular direction, it has a definite rate, and it could be altered by certain conditions. So these chemical changes together only, you call that as a metabolism. So the metabolism is all the chemical changes in the living world. What is common? These metabolism and these chemical changes are called metabolism. Because this is essential when you go to the enzyme in this chapter. Now chemical changes are known as metabolism. Metabolism are metabolic reactions. Metabolic reactions. Okay. Now, these reactions, very important, these reactions have a definite pathway. And it has a definite pathway. That the metabolism has definite pathway. Pathway means direction, and it is occurring constantly. Constantly means continuous, constantly occurring reaction. Then it has a the substance is produced has got a definite role in the body function, building up of the body, or activating the body, or producing energy. So they have a definite role. Second, they have a definite role in the body, definite role or definite function. What they have, the product has got definite function, and they have a certain rate or speed. or you can say they occur in a certain speed, which could be altered. It can increase or decrease. So the speed can be increased or it could be decreased. So these are so they came to the conclusion all these chemical substances in the living body. So these chemicals which are found in the living body called that as the biomolecules. First, they compared living and non-living. They found out equally there are many elements present, but mostly carbon is abundant only in the living tissue and water is a major abundant one. So those chemicals mainly present in the living world, you call them as the biomolecules. So the bio, living, molecules, chemicals. So of these biomolecules, they wanted to differentiate into you cannot generally say everything carbon, hydrogen, nitrogen, oxygen, sodium, potassium, salt, everything is a biomolecule. They are. There are all their chemistry, what they are studying, metallic and non-metallic elements are all involved, except the higher isotopes and the rest of the things. All the basic elements are involved in the living, present in the living tissue. But these substances should be categorized, grouped. To know why should you group it? To know how is it present? Why is it present? What is the effect of it? Is it doing any work in the body? Or is it of any use to the body? So they wanted to study the biomolecules further. What is biomolecule? Simple word. Chemicals present in the living tissue. That's all. That doesn't mean they are absent in the non-living world. They do present in the non-living world also. And these elements, particularly, they form some of the compounds, but particularly carbon is present in all the living tissue. That is why you study carbon as a different group in your chemistry as organic chemistry. It's a vast one equal to all the compounds in your chemistry put together is equal to your carbon compound. How in animal kingdom? Animals in the arthropoda is equal to all the animals in the different groups put together, phylum put together. Same manner, carbon is a vast group. It contains all the chemicals together put in one section, that is carbon. And you have a separate study in chemistry for carbon alone. You call that as an organic chemistry. Why? Because carbon is present in the living world. Organ belongs to living tissue. Then they wanted to see the biomolecule, find out the biomolecules. Now, why are they interested in why if the body is made up of the biomolecules? So what is the use of this molecule? And how does it help the body? Is it building the body or is it activating the body? Is it undergoing? And all these things, the question came. Then they started analyzing the biomolecules. How will they analyze the biomolecules? Simple test. Remove the water is abundant, remove the water by dehydration. Then they will get an ash automatically. When it's burnt, in simple language, it's burnt. So when you get the ash, that ash cannot be salt form. You dissolve it in the solvent, acid, which will dissolve it easily. Particular acid you take, in a textbook will be given hydroxychloride acid will be given, and that is dissolved. Mixture is filtered, and when you filter it, some will be on top of the filtering membrane, another will be coming down. Some dissolved. So those which dissolve will be filtered as a filtrate, and those undissolved matter will remain on top as a supernatant on top. So residue on top. So they took separately. The liquid they took separately and found out what are all in the acid soluble substance and what are not acid insoluble. They categorize biomolecules as acid soluble and acid insoluble. Okay. Now, so then that is whether filtrating unfiltered. So they divided that into acid soluble, those which dissolved in the acid, and insoluble, acid insoluble, that is undissolved. Why they are soluble and undissolved? Automatically, now these are the size of these molecules are smaller. Now these are larger molecules which cannot be filtered, and they are smaller molecules which will easily dissolve in the medium, in the solvent. So their first differences they found out, they have difference in their molecular weight. The molecular weight in biology, what is atomic weight, atomic mass unit which you consider in chemistry, is it is written as some books it become Dalian or Dalton. Dalton is the same as AMU, atomic mass unit only. So the atomic mass which is represented in chemistry, in biology, you call that as a Dalton or Dalian. So symbol is Da. So they found out why they are solved. Main difference is one is larger, heavy, One is lighter, so lighter one dissolves in the acid, and the heavier one remains on top. So they separate the unfiltered one separately, analyze what are the chemicals there, and the filtered liquid they took out that, and then analyze what are the ones which dissolved in that. Then they have analyzed that. They come to the conclusion. Some of them are they undergo a specific chemical reaction, a chemical reaction in the body. Yes, sir.
Continue.
Chemical reaction in the body. So these chemical reactions, you call that as a metabolism or metabolic reaction.
Some shaking.
Pardon?
Some shakings.
Okay. A metabolic body. So they found out some of these chemicals undergo definite chemical reaction. And this chemical reaction is not a haphazard one. It occurs regularly in a particular part of the body, in a particular cell. Body is made up of tissue made up of cells. So these metabolic reactions are specific. They are definite. They occur in particular cells, and they have a definite direction. And the products or byproducts are produced, and the products are useful in many ways to the living body. So without the chemical reaction, which provides the energy, the body which is built up of these biomolecules cannot survive. This gives you another concept in your ecosystem or biosphere. The living world is made up of matter and energy. What is this energy? To matter is a simple model. Suppose you are making a car model for your project, and it is a dummy model to keep in the showcase. Suppose you put a battery, it will become an active model. That's a human body. Without the energy, I am a dummy. So without the body completely is made up of these biomolecules. But these biomolecules simply made a dumb body or an inactive body. It's of no use. So it has to be provided energy to activate that energy back again by the chemical reaction. Energy is come from the sun is the only source, but that solar energy is trapped by these chemicals, and the chemical and the energy is transformed into the chemical living world, and it provides the energy which is needed for the living tissue to activate. So they found out after analyzing their micro and macromolecules, they found out that is okay. Now, 1 second. They found out these activities have got a definite pathway and they have a definite role to play. So those substances and there are some substances which do not undergo chemical reaction. Why are they present then? They build the tissue. They are necessary to build the tissue. So they're building material. So in a house, everybody need not be active. One person can be active. Father may be active and mother may be doing another kind of work, not activities there. But these, what is the function of the other ones which do not undergo the chemical reaction to produce energy? They get involved in the building up of the body. So the substances or biomolecules which are actively taking part are known as the metabolites. Why are they called metabolites? They take part in the metabolic reaction. What is metabolic reaction? To understand, they're nothing but chemical reaction in the body, biochemical reaction. All right. So they are called primary metabolites. Now there are in plants, they found out apart from this primary elemental level, there are certain substances produced in the body of the plant, mainly in the plant. Not in the animal also, you can say like certain insects, but another higher order of animals. So when they analyze the plant, not only plant tissue, they started analyzing the fungal tissue or microbial tissues. And when they analyzed the microbial and fungal and some of the plant tissues, they found out apart from these primary metabolites, there are certain substances present in the plant body, which of no use to the plant. It is of no use in the plant in a sense. It is just produced in the plant world, but it is used for the animal world. For example, gum, resin, rubber, latex, some alkaloids or drugs, oils, scents. All these are the excretions of the plant. Plant oil, grass oil. Now, this one, alkaloids, many drugs, cocaine and codeine. Now, certain, say, rubber, latex, and all these substances which are produced in the plant world only. They do not undergo chemical reaction. They are not involved in the production of energy or maintaining the body. Even if they like cell inclusions, if they are or not, it doesn't make any difference for them. But it is produced in the plant world, which are not much used to the plant, but it is much used to the animal kingdom. That we make use of the drugs, we make use of the oil, we make use of the rubber, latex, etc. So they are called secondary metabolite. Primary metabolites are the chemicals. After analyzing all that add is insoluble, generally they came to the conclusion these biomolecules, some are metabolites, some are primary metabolites, some are secondary metabolites. All right. Now see, after this we'll go to the next section. The next what you have to see. So you follow up along the biomolecules first. In a nutshell, biomolecules. Then biomolecules or under biomolecules, metabolites. Metabolites. Then that they are called, sorry. Primary metabolite. And secondary metabolites. Secondary metabolites. Alright. Then how do they analyze these biomolecules? The biomolecules are the primary metabolites are again, there is A and B. These primary metabolites can be again grouped as the macromolecules and biomacromolecules, macromolecules and bio, that is bio, this is bio, micromolecules. Micromolecules. How did they find out? By filtration. So what is the main difference between them? In their weight. Alright. So biomolecules can be divided into primary metabolites and secondary metabolites. Primary, we can have a flow chart like this way. See BIOM molecules, primary metabolite, met, and secondary sec metabolites, and primary are divided into acid soluble and acid insoluble. So they consider these as the biomacromolecules. Alright. Now what is the character? The biomacromolecules, they are all polymers. You know very well what is polymer. Polymer is a group of, so don't use the word molecule or atom. It's not a group of atom or group of molecule. It's a group of simple similar units. So you should use the word polymer is a large unit. Unit is the word you should use. You should not use the word molecule or atom. Formed by smaller similar unit. Similar or dissimilar unit. Okay, I want to use the word similar. It may not be always similar. So, smaller units. Okay. The smaller units are called monomers. Alright. And another importance of this acid soluble ones are in their weight. Now difference is they are heavier and this is lighter. Their weight is about 10,000 and above Da. Da means Dalton or Dalian or AMU. Da or Dalian or AMU. Alright. Now these ones 80 to 800. molecular weight, DALIA, BA. So they are soluble in water, they are filtered. So we are now anchoring on the biomacromolecules. So what are these biomolecules? Now they are polymers. They are made up of smaller similar units of monomer. They have got a weight higher than that about all those molecules which are above 10,000 molecular weight. The weight is AMU is above 10,000 DALIA are grouped under one category. All those which is lesser than that automatically they get dissolved in the acid. They are acid soluble. They are separated. So in this, when they found out this acid soluble, so macromolecule and micromolecule. So the macromolecules are back again acid insoluble. Okay. Now the first character, they are polymers. They are polymer. Then what do we say? They are primary metabolites. They are primary metabolites. Second character. Primary metabolites. What do you mean by primary metabolite? They undergo a definite chemical reaction. The chemical reaction has a definite path, an intermediate product, starting and the beginning and the ending. It starts with one chemical and ending with the other. And during the reaction, byproducts will be there, and energy will be released, and useful substances produced, and those useful compounds can be used for building up, energy can be stored or utilized, and the byproducts are the way, if it is waste, it is excreted. So within this chemical reaction, all your other systems of your excretory system, your circulatory system, all depends on that. Major your excretory products are coming from where? By the chemical reaction of the metabolism only. That's why if you remember, I told you excretion is excretory matter is as a result of chemical reaction. And digestion is not as a result of chemical reaction. The undigested matter is digested, not excreted out of the body. Alright. Now, when you come to this, now we are going to this. So these are called the biomacromolecules. So these are the biomacromolecules. Now biomacromolecules. Now, how are we going to study this? So now we'll stop with this itself. Now under this, what are the Neet point of view questions can be? Okay. Now first, they'll ask you what biomolecules are. They want to ask you what is biomolecule. That will be like a lower class question. They will give you macrobiomolecules are, or they can give you carbon, hydrogen, and the element mixes up. When it comes macro, you should fix up only carbon, hydrogen, nitrogen, and oxygen. Other things are micro. Then next question, the acid soluble molecules are. They can give you back again all the major ones. The macromolecules will be acid insoluble, the other one will be soluble. All right. The next one. Now primary metabolites are. That's very important. Primary metabolites and secondary metabolites. More than primary metabolites, invariably secondary metabolite will be asked. Now the secondary metabolites, two questions will be asked. Secondary metabolites are found in, they'll say animal tissue, plant tissue, or microbial or fungal or none. That's one way. You should remember secondary metabolites, they mainly some plant in a sense, what I meant, in a sense gymnosperms. You know very well, botany gymnosperms are the conifers, and most of the oils and perfumes and scents are produced in that. Now, what is this oil and produced in those plants? If you remember, the oil is in during the second, the inner, the core, hardwood, will be trapped in the tissue. Wood formation, secondary thickening, and the wood formation, this oil and extract will make the central core more stronger. So it is a product, but it is not of any chemical reactive value. But it is produced in the plant. So these things ooze out. Latex is nothing but a secretion. And like a gum and latex, a secretion, lactiferous cells. Now these cells, the latex is not used by that, it oozes out. And we extract that and make out rubber, gum, resin, scents, perfumes, oil. So they'll give you secondary metabolites are found in animal tissue, plant tissue. If they give fungal and microbial as well as a plant, choose only the fungal and microbial. Major analysis is from there. When they say plant, now they are not talking about the angiosperms, they are talking about gymnosperms. Remember that. So then none. The first simple option. Second option they will give you. Among they'll group up 2, 3. Okay. Now they'll give starch and gum and protein or amino acid, and one rubber latex, and they'll give you one pair with the two plant oil, that is scent perfumes and alkaloids. Alkaloids are mostly drugs, which you will study in 12th standard on the particular chapter. Completely, what are the drugs, what are hallucinogenic drug, psychedelic drug, and psychotropic drug. So these drugs analysis to all this atropine and medicines like atropine and belladonna, all these mostly gymnosperms, they produce is alkaloid drugs. So they will give you pair by pair by pair. One will be a normal product of a carbohydrate and latex rubber. One of the pair will be correct metabolite. Maybe gum and resin or scent and plant oil. Plant oil, example, cinnamon oil. That is one of the product secondary metabolite. Okay. Now these things, second question. First is oriented on secondary metabolite, how do they get? Second, what are the types? Find out which is among this following, which is the secondary metabolite, which pair represents secondary metabolite. That's one question. Now, third way they can ask you what? Now, the secondary the primary, the main difference between the primary metabolites among the metabolites, okay? Now, the one which undergo metabolic reaction, which undergo metabolic reaction and non-metabolic, no metabolic reaction. In that, they won't give you primary and secondary. There will be two choices only in that case. You know very well, primary metabolites undergo chemical reaction, secondary metabolites do not undergo chemical reaction. How the question can be worded? I will use a molecule which is not a secondary. For example, carbohydrate and protein. They'll give you among the given statement, which pair or which are. If they give you single option, say for example, another question. Okay. Fix up which among the following list given, which are primary metabolites, which are secondary metabolites. In that, they will give you only one question. Two questions I can ask you. I'll give you an option, 4 options, directly, and ask you among the given list, which is the primary metabolite, and among the given list, which is the secondary metabolite. All right? Do you understand what I'm telling? So you have got 2 lists directly picking up. So primary metabolites, what will I do? I will give you carbohydrate, protein, and carbohydrate, only one, or they can give pair by pair. One primary metabolite I'll give you, rest of the thing I'll give you is a salt, a perfume, or rubber and latex. Similarly, when they give the secondary metabolite, they'll mix up the same question can be mixed. There's only one secondary metabolite, rest are all the other chemicals. So the secondary metabolite is an area in the introduction part, which can be asked in any manner in the question. First part of your lesson. All right. Now the another one they can ask you, that is, now which of the following molecules in the given list undergo definite metabolism or definite chemical pathway? If they say metabolism, you easily find out. So undergo definite chemical pathway. If I give the word definite metabolism, means you will immediately know, you will sort out primary metabolite and secondary metabolite. If I give you starch and the rubber, automatically you will take out starch and you will leave the rubber. So they'll ask you which of them undergo definite chemical reaction in the body, in the living body. Which you should immediately, which will undergo the chemical reaction? Primary metabolite. What is the primary metabolite? That's a mostly in the animal tissue. That's carbohydrate, protein, fat. And the other things are, so you have to fix up according to that. Read the question. Options will be very simple. The fact is very simple, but they will wording will be twisted. So this is the first part of your lesson. First part of your lesson. What did you know? Living and non-living matter. Comparing, there are chemicals in the living world. They call that as the biomolecules. Now, these chemicals in the living world can be categorized. They want to analyze the living tissue. When they analyzed it, they found out the different types of molecules. Some are heavier, some are lighter, some are soluble in acid, some are insoluble in acid. And they found out that some are in the salt form, some are in molecular state, carbon, hydrogen, nitrogen. and some are in the salt form. So they found out some biomolecules are in the molecular state and some are in the salt form. Salt form means basic and acidic sodium carbonate, sodium chloride. That is salt form. Then next what they found out, these molecules, biomolecules, some are acid soluble, some are acid insoluble. Third point, some are heavier in molecular weight, some are lighter in molecular weight. Fourth point, some can undergo chemical reaction, some do not undergo the chemical reaction. So these are the analysis of biomolecules. So then they group the biomolecules as a primary metabolite and secondary metabolites. Next point. And then they grouped it as a macromolecule and micromolecules. Bio-macromolecule, bio-micromolecules. What is the difference in a molecular weight? 10,000 and above Dalian macro, 8 to 800 Dalian micro. Lesser molecule dissolves the acid, higher one will not dissolve. So what are these? In this lesson, you are going to study about the biomolecule, macrobiomolecule. That's the second part. Okay. Now the second part of the lesson is macrobiomolecules. They are macrobiomolecules and now first point you should know, they are first point is what? They are primary metabolites. Remember that. They have undergo chemical reaction. Second, what you know, they have got heavy molecular weight. Third, what you know, because they are undergoing the primary chemical reaction, they are involved in the building up of body, or producing energy for the body, and producing the waste byproduct during the reaction. Please don't forget that. One is useful substance is produced, waste is produced, energy is produced by this chemical. Any chemical in the biochemical reaction in the body, biochemical reactions, three are the major principles. One is energy will be released. In any chemical reaction, you know very well, energy is released as exothermic, endothermic, heat energy is absorbed, and heat energy. So energy in one form will be released. So the energy is an abstract term. You know that energy is nothing but a force or a capacity. So this force you cannot collect. How will you collect it? It could form a binding. That is bond. What is the bond in chemical reaction? Bond is nothing but a force which keeps two elements together. Either it is an electrovalent or covalent. So bond is nothing but a force binding. That's why the word comes bond. So the energy is a force which cannot be collected practically like a chemical, like a CO2 or H2O. So the energy should be used to make a bond, used to bind certain elements or certain molecules. So any chemical reaction. So the macromolecules have got major. Don't forget this point. Macromolecules that have the molecular weight heavy. They undergo chemical reaction. In chemical reaction, you call in biology as metabolism. This chemical reaction only, the word used as metabolism. And what is the use of the metabolism? A useful product. Any chemical reaction, there are reactant and product. So the product here is a useful product which will build up the body. Useful product, one, building the body. Then second one is a useful product. Similarly, byproduct, unuseful. Unuseful ones are the waste. It should be eliminated out. Sent out, your body cannot accumulate the waste. Thirdly, production of energy. Energy release. Energy produced. This energy produced in your body is used in three ways. The energy can be used by the producer, that is by the structure in which the chemical reaction occurs, or by the body, in other words. So the energy is used by the body which undergoes this reaction. Secondly, energy is used by the body in a sense, it is used by the body for its function, for its activity. Suppose take your body, energy produced in your body is used by your body, not only to do the work like walking, talking and everything, but it is used when even when you're asleep, your inner organs have to work, your heart has to work, your stomach works. heart, everything, the brain works. So the energy produced in your body is automatically used by the body in an when you're awake or when you're asleep. Every living organism is the same. So the part of the energy is the body. So part of the energy is building up the body. How energy can build the body? Your body, say for example, your body is made up of protein. What is protein? It's a polymer. How will you produce a polymer? Combining acid. How will you combine the acid? By the bond. What is bond? Energy. So the energy used to build the body by preparing the raw material. What are the raw materials? Can be carbohydrate, fat, protein. So that's why simply we say energy used to build the body. Thirdly, excess of energy will be stored in the body. How is it stored? I told you energy is an abstract term. It's a force. It can form only a bond. That's what I mean by storing. So it is stored as bond. Particularly a chemical in your body is adenine. Adenine. It's a nitrogenous base. That's what you come across AMP, ADP. Adenine monophosphate. You combine adenine and phosphate, you form a single bond. That bond is the energy. Adenine diphosphate, triphosphate. You follow? So the energy produced by these are the properties of your biomolecules. The most important property is they are the polymers. They are polymers. Important one is polymers. We are only focusing on they are polymers. What are polymers? Larger unit made up of smaller similar units. That's all. It is the smaller should be the similar. Similarity should be there in one way or other. Sometimes it can be dissimilar things also can be combined together, but polymers are the larger units made up of smaller units. The smaller units are called the monomer. That's all. Now, what are we going to do in these parts? We are not completely chemical reactions we are studying. What are the chemical reaction? In photosynthesis, C3 cycle, C4 cycle, citric acid cycle, glycolysis, and chemical citric acid cycle in the cell, RBC, yeah, mitochondria. So all these metabolic reactions goes on continuously in your body. And if there is no metabolism, the body is dumb dead. It's a cadaver, inactive. There is no life in it. So there should be always this metabolism is there in your body. So these metabolism can be breaking down process. If you store the energy by a chemical reaction, you are producing a lot of energy. Energy is available, you are storing it. Then anabolic part property. When you are breaking, automatically anabolism and catabolism. Anabolism is building up, catabolism is breaking down. Both are like oxidation reduction. When one is broken down, it will be producing something else will be produced. Say for example, glucose is broken down, catabolic. But at the same time, it is building up the ATP that is anabolic. So catabolism and anabolism will go hand in hand together in most of the chemical reactions. You follow? So now see these are the aspects. The molecular weight we already saw. Metabolism. Now, second part of your lesson, you are talking about this metabolism, how this metabolism, not about the pathway of the metabolism, how this metabolic rate, what it is, or what is the rate, how it is enhanced, it can be increased or decreased, mainly the presence of biocatalyst, that is why the enzyme next clicks on the metabolism. Now, next one is usually the energy produced, we are learning actually in the chemical reaction. Then useful and waste. Waste is near the excretory system. Did you follow that? So now let us go to the formation of these biomolecules. The second part, the third part of your lesson is the macrobiomolecules, since they are polymers. Polymers. Now first, before you go to the, what are these major macromolecules? The major macromolecules in your body, they found out mainly carbohydrate, carbohydrate. I should not use the entire board. Yes, you can see the word angel. Carbohydrate, carbohydrate, then protein, and fat. Now these are the major macrobiomolecules. Alright. Now in this, the fat is an exemption because these two have got a heavy weight. Heavy double, that is they are 10,000 and above Dalian BA. But this one, the fat is below 800 only. 800 Dalian. Yet it is considered as a thing what it is because these are, what are these things? They are unfiltered. They are undissolvable, acid insoluble. Both are acid insoluble. This is though the molecular weight is less, it is acid insoluble. It cannot be filtered. Why it cannot be filtered? Though it is a smaller one, it is also acid insoluble. Now, why acid insoluble they are? Because this fatty acid does not occur, if you know in a cell, cytoplasmic pool is there. So the fat acid is not fatty acid, fat acid gets involved in the constructing the different parts of the cells. For example, it becomes a component of a cell membrane or membrane covering the organelles, or it is becoming a building material within the cell. So how much ever you smash it out before dissolving in the acid, it does not get broken up into tiny molecule which can dissolve in the acid. It forms small bubbles or globules, vesicles, you call that as small bits and pieces, vesicles. These vesicles do not filter down. So you group them under the bigger item which is not acid insoluble. So why is it not, although it is acid insoluble? The reason is it forms, it becomes a structure of the cell, structural component. The word will be given in your book, structural component. What is meant by structural component? That is such as it becomes a part of cell membrane. Example, it becomes a cell phospholipid, cell membrane, or a membrane of the organelles, building up of the organelles. So when it becomes a structural component, it doesn't break down. You cannot be smash it out, or you cannot powder it down, or completely. You cannot. It cannot be broken down into a substance which could smaller material which could dissolve in the acid. They form bubbles and vesicles. They form vesicles. Fine vesicle means small globular structures which do not dissolve. So it is along with the unfiltered area, unfiltered substances, it cannot be filtered, it remains. So that is why fat is considered along with the carbohydrate and protein and fat. Otherwise, structural form, it is completely more heavier than the other two. Okay, now these are the macrobiomolecules. First point, they are polymer. So what should you need? You need the monomer. Okay, now this is the the third part of your lesson. Now we are going the monomers. First, how will we study? First, we study the monomers, different monomers of carbohydrate, of protein, then our fat. When these monomers, how are they going to? Second, we will study how these monomers are joined together to form a polymer. What are those polymers? And what are the uses of the polymers? How do they form the bond? That is second part. They follow. So when that, with that, you will the biomolecules will be over. So the macrobiomolecules. The next part we have to see completely about the monomers of all this. Then now, so the next portion is your next slot is your monomer. Now we will take the carbohydrate alone. First, you take carbohydrate. Okay, carbohydrate is a polymer. The monomers are known as sugars. Monomers are sugars. Now the sugars are simple sugars and compound sugars. More than one sugar combined together. The compound form. So why I say compound form? Because you can split it. You know very well. It's like we cannot say the word in a physical. On a chemistry level, you know very well compounds and mixtures, but we cannot call them as a mixture. They are joined by a bond. And again, the bond is broken, they can come back to the monomer. So the monomers are the sugars. The sugars are the simplest sugars that are called monosaccharides. Sugars are saccharides. Saccharides. The simplest of them, or the simplest one, is the monosaccharide. Monosaccharide. And there are three monosaccharides. Namely, one is called glucose, the molecule. The next one is fructose. Another one is galactose. Before you go to this fructose, galactose, all these carbohydrates are chemically hydrated carbon. Hydrated carbon. What do you mean by that? The empirical formula CnH2O(N). This is a empirical formula. Say for example, if I have, for example, C5H2O5, that means what? C5H10O5. Alright? So this is the C5H2. That is CnH2. How many ever carbon atom is there? So many ever water molecule. That's all. That's why the word comes carbohydrate. Car stands for your carbo, stands for the C, and hydrated means H2O. Okay. So this is one. If I want to write this as a structure formula, every carbon atom has got one hydrogen atom, and see, you know very well water can be written as in the bonding. Polar covalent compound. You will write it like this. So the same thing in this it will O C in the middle. The water is split as OH and H, and the H on one side, H this side. You know very well carbon has got 4 valency. So to this valency something else can attach. To this valency something else can attach. But in every carbon atom you have one water molecule sticking on to that. That is why the word is carbohydrate. So the carbohydrate substances. Now, when you talk about hydrated carbon, now glucose. Second thing, what is according to the number of carbon atom. According to the number of carbon atom, they are named as these carbon compounds are called one carbon compound, 3 carbon compound, say 2 carbon compound, 2C compound, 3C compound, 4C compound. Now, say for example, 2Z. What's the thing? The right you have got the pyruvic acid, 3C phosphoglyceraldehyde. Now 4C oxalic acid, 5C ribose sugar. Now 5C is ribose sugar, and 6C is glucose. But mostly these glucose, fructose are 5 carbon sugar. Now these are all 6 carbon, 5 carbon compound. You call them as a 5 carbon sugar or 6 carbon sugar. So definitely you will have a structure for that. So what will be the glucose structure? You know then we'll go to the structure. So the monomers are sugars, and the simplest they have got the simplest form of monomers are called sugars or saccharides. The simplest is a monosaccharide, and the other ones are disaccharide. When they form the polymer, it will become a di and tri. Polymerization is not a chemical reaction; it could be broken down. Breaking up of the polymer is known as the cracking. You know that in chemistry. So when they crack the bond, it will become a monomer. When you join the bond, it will become a polymer. So the monomers or the simplest form of the monomer of the sugar is actually glucose, fructose, and galactose. Now they can form polymer. The polymer may can have more than one sugar. It can have 2 sugar, or it can have tripolymer, tetrapolymer, pentapolymer, heptapolymer, or polymer more than 3, called that as a polymer only. So 2 carbon compound, 3 carbon compound, trioses, trioses, tetroses, pentoses. The 5 carbon is pentose sugar. Tetroses 4 carbon. Heptoses 7 carbon. There are 7 carbon compound also. So according to the number of carbon atom, you will name them. Now when you are talking about the simplest, there are only 3 basic building blocks: glucose, fructose, galactose. They are in the combination, they can form a number of polymer. It can be glucose, a number of glucose, 2 glucose, 3 glucose, 4 glucose. If 2 glucose bonded, it will form one sugar. If 3 are bound, another sugar. If 10 are bound, another sugar. A number of glucose acids are found, joined together, bound together. It will form another substance. You name that as a polymer. It is a polymer, but it has got a particular name. For example, cellulose. Cellulose is a long chain of number of glucose. Now, starch, same like cellulose, but the arrangement, structural formula will change. A polymer, like isomerism. Isomerism, same molecular weight, the structural formula we have, no. So, in this case, isomers, the structure will be the same, structure will be variant little bit. So, now the major what you should understand, the major monosaccharides are glucose, fructose, galactose. Understand? Now, carbohydrate is the first biomolecule, important, the most important one. It is an energy source of energy, calorific, but the calorific value of the carbohydrate is slightly lesser than fat. But the molecular structure, the fat is more complicated, so it cannot be broken down. You will see the bond formation, then you will know how to break it. So the bond, unless you break the bond, energy will not be released. What is metabolism? Chemical reaction. What are you doing? Breaking. What are you breaking? Breaking the bond. So polymers are broken down. During the breaking up, energy is released. So now the monomer of the carbohydrate is known as the sugar or saccharide. There are simple one sugar is known as monosaccharide. There are 3 basic monosaccharides. You follow? Now next one you have got to. Then another thing in this issue, the carbon, the compounds can be named according to the number of the carbon sugars are named according to the number of carbon atom in there. That is 6 carbon, trioses, pentoses, tetroses, hexoses, hexo sugar, glucose is a hexo sugar, ribose is a pentose sugar, heptulose, 7 carbon compound. Now 4 carbon compound, it need not be always OSC sugars. It can be an acid, oxalic acid, and 3 carbon phosphoglyceraldehyde, the 3 carbon compound. So all these things are named according to the number of carbon. We are not bothered with the number of carbon compound. We are only bothered about now the glucose is 6 carbon compound. Now the fructose and galactose. These are the 3 major ones. When you talk about next, we will go to the monosaccharide. Okay, these monosaccharides in turn. So monosaccharide there are 3. That is fructose. I'm sorry. Started the glucose. Glucose, fructose, and galactose. It's a six carbon compound, C6, the C5. Now listen, back again C6. Now next slot what you're going to do, now disaccharide. I'll write it as MS. MS means monosaccharide. Okay. Now then next you're going to the disaccharide. Now disaccharides are any of these 3. That is, if you consider A, B, C, A plus A, or A plus B, or A plus C. So it can be glucose plus glucose. You call that as a maltose. It's a maltose. Now glucose plus fructose. You call that as the sugar or the sucrose or the what is sucrose is the sugar that you use in your coffee. Now fructose is the fruit sugar and the glucose plus galactose that is lactose and only one disaccharide for fructose plus fructose is a peculiar one, inulin. It will give you inulin. Now inulin is a disaccharide. Don't muddle up the word insulin. Insulin and inulin. Insulin is a hormone protein compound. It's a protein substance. Inulin is a carbohydrate substance. So the bond here is different, there it is different. Now these are the disaccharides. Next you go to the polysaccharide. Poly means what? More than two. Poly. Example, Pakka example you will take cellulose. Cellulose, a basic one, where you have got a number of glucose. This is one glucose and bond. So the glucose units are, say for example, cellulose is a fiber-like structure. Now there it has got a straight chain formula, straight chain combination. Glucose, another unit of glucose, it goes on. Bonding. It will form a long thread-like structure. Another thread-like structure, another thread. This is glucose. I'm sorry, this is called cellulose. Now the straight form, suppose one thread is there, that it is going on straight, it will have a branching here like that. It branches, but all the branches are not coiled one straight. Now this is nothing but starch as well as glycogen. They have a branch, the variant of cellulose only. Basically, all these glucose, many monosaccharides are attached in a straight formula, straight manner. So it forms a fiber. The substance which has got the maximum number of cellulose is the cotton fiber. Cotton fiber is the one which is fully composed of the polymer cellulose, which is a polysaccharide of the glucose. Only glucose unit is there. Now, when you are forming this bond, see here, it's glucose plus glucose, and this is the fructose. So this can be homopolymer. If you go on adding glucose, finally you will get a cellulose or a fiber. So when the polysaccharide, many units combine together, the product itself is changes. See, if you, for example, maltose is a sugar, malt sugar, which you can dissolve or eat. But when you talk about the cellulose, you cannot be eating the fiber. You know very well, there is no enzyme digesting cellulose in your cellulose digesting enzyme in the human body. Cellulosin is present only in the herbivores. So now when the molecules combine together, the nature of the product itself will change. They follow that. So now this is polysaccharide. Difference, the structure, cellulose is a basic polysaccharide, common polysaccharide. Now there in variant it will be, the variant means slight difference in your structure is a starch and glycogen. In that also, the starch branches, the branches will form, say there is another branch and you have another branch, it might form network. In the network, iodine droplets will get caught, I2. That is why the test of starch, when you add a drop of iodine, the whole thing becomes blue because of the network can hold iodine. But in the glycogen, the network is there, but it doesn't hold iodine. They follow that, and you know very well, cellulose is one of the major building substance of the cell wall. Okay, now see, now these are the different polymers of glucose. Now you should know the structure of this, the molecular structure of the glucose. Now glucose is a 6-carbon compound. See, molecular structure. You can now up till this, what questions can I ask? We will finish this glucose alone. Now, if you take a glucose, it's a C6 compound. So naturally, you know very well, it will have a hexagonal form. Yes, every corner represents, you know very well, every corner represents the carbon in the carbon atom. In this case, this is first carbon, this is second carbon, now third carbon. structure of structure. When you come to the structure part of it, now you take the glucose, structure of glucose. Glucose is a C6 compound. Therefore, the molecular formula should be what? C6H2O6. So that is equal to C6H12O6. All this you know. How to put it up in the molecular figure? It's a C6 carbon. The figure is as a hexagonal ring. It's a ring, closed ring. Then this hexagonal ring, each corner we have a carbon. There's one here, 1, 3, 4. So if I join this will be the angle. Every corner you have got a carbon. Now in this is carbon, here is a carbon, here is a carbon, here is a carbon, but there is no carbon here, it is O. And the next one will be attached to the next carbon. I take it like this, please. Now though it has got 6 corners, now first one, this is first carbon, second carbon, third carbon, so fourth carbon, then fifth carbon, sixth one we attach the fifth. So this is O. How to remember this formula? Now everywhere you have got to see. Excuse me, I'll draw a little bit lower so you will know the structure. 1, first carbon. This is carbon 1, carbon 2, carbon 3, carbon 4, carbon 5, 6. Now every carbon has got HOH, water. You know it. So here you have got HOH. Now similarly it can have HOH here or OH here, H. Then this has got now HOH. Now this fifth carbon will have a HOH. Now let us take the fifth one. Now fifth one is connected to the sixth one. So what will it do? Is one of this O will come over here. The fifth carbon's O group comes here. So one of this hydrogen will remain attached down here. Hydrogen will be there. But the other hydrogen will go up. So when the other one is pushing up, that has already got HOH. This H will become 2. That is why the sixth carbon, if you see, it only HOH is the H2OH. So usually when you are drawing the figure or when you are looking at the molecular structure, it will look alike as everything. But you should know how is it at the fourth, fifth carbon. Suppose if it's a fourth carbon, same thing comes in the case of the ribose sugar. In your when you draw the DNA structure, ribose sugar, bonding between the sugar and the base and phosphatide ester bonding. So in that case, you should know how to draw that. So suppose this is a fourth carbon. Now fourth carbon has got one O and one OH. Now fifth carbon also has got one OH. So fourth carbon donate the O here and donate this O is gone. Now this H is remaining here and the other H is pushed upward. That to go to the next carbon. Next carbon already has got a H2, so this will become H2OH. That is why the sixth carbon will have a different. Rest of them are now. This is fifth. This is sixth. So the fifth will continue as the fourth. It has to go to the fourth to third, 2, 1. It will close up like that. You follow? So this is a fifth one, fourth carbon, third carbon, second, first. Now same thing you will have in your ribose sugar. Suppose it's a 5 carbon sugar is a ribose sugar. So how will you draw the structure of ribose sugar? So ribose is the first ribose. Let us take ribose sugar. Ribose sugar is 5 carbon. Let us C5. So what will be the formula? C5H10OH. Now how the figure will be? This is a hexagonal form. Now this will be pentagon. Like this. So first, second, third, fourth, fifth will be attached to that. So fifth one will always have CH2OH. This fellow will have only H. And this will have OH, HOH. Then HOH. This also HOH. This is O. Invariably, when these sugars come, this is a ribose sugar. This is glucose. 5 carbon sugar, 6 carbon sugar. You know very well, the glucose is joining with the another glucose. Now adjacent glucose, suppose if I draw glucose 1 here, the next glucose, the fourth is coming near the first. So the connection will be from 1 to 4, 1 to 4, 1 to 4, 1 to 4. Suppose I am using a number of glucose. I have to join a number of glucose. I am putting all these things side by side by side. Say 1. This one. Then another one. What is this one? This will be fourth. This will be first. This is the first. So the connection will be 1 and 4. Now back again, this will be 1. And the next one will be 4. This will be 4. So the connection is 1 and 4, 1 and 4, 1 and 4. Bonding. That bond between the glucose units are called glycosidic bond. Now this is called glycosidic bond. Glycosidic bond. Now before we proceed further, let's see what are the questions I can give you within this part, how much ever we have discussed so far. Alright? Now see, we went for the first part, we finished. Then we have to take the polymer. That is a polymer of carbohydrate. We are going to study about the carbohydrate. Now carbohydrate, you know very well, it is a polymer. The monomer is a sugar. And there are only 3 monomers. So how the question can be? Now they can give you first the monomers of carbohydrates are direct option. They won't give you glucose sucrose now. They can give you glucose sucrose to Madliap. If they say monosaccharide, easily you'll find out. So what I will give you, the monomer of carbohydrate, the basic monomers are, instead of giving monosaccharide, I will give a name of glucose, fructose, galactose, amino acid, or lecithin, or cellulose. So I will post the name of it. I won't give you di or poly, so you will easily find out if a single word is that's a monomer. So I will use the word the monomer of a carbohydrate. Only one answer should be correct in your option. And that will be glucose. Second option is galactose. Third option is inulin. Fourth option is cellulose. It need not be always carbohydrate. You can make anything else. Inulin you can lecithin, that's a protein compound. Polymer. So any of this organic compound I can write. So you are expected to fix up. You should know which of these chemical name is a monomer. So first question can be the monomer of carbohydrate. They won't tell you monomer is a saccharide or monosaccharide anything. They'll give you the name of the monosaccharide and other compounds. You have to pick up that. That's one option. Then another thing, the disaccharide name they'll give you. Formed by disaccharides name and now the disaccharides of glucose. They will give you inulin is not a disaccharide of glucose. Any one of these disaccharides they can give you maltose or sucrose or lactose. Alright? Or disaccharide, any one of the polysaccharide they can give you. Or a dipeptide they can give you. Glyceride. So any of the biological name, molecular name they can put up. So you should know which is the disaccharide. Which are the disaccharides of these are the disaccharides so far what you are learning. Another important one, the disaccharide inulin is a disaccharide of galactose, glucose, fructose, glucose, galactose or cellulose. You should know there's only one disaccharide for the fructose, that is inulin. Okay, now the next question they can ask you. Now the formation of, they can mention the substance. The basic units present in sucrose are, the basic units in sucrose are 2. Till muddle of this. You should know glucose and fructose forms the sucrose. In another word, they can ask you one more generally about the sugar which we use, the sweetener, the sugar. The sweetest sugar is called levulose. The smallest and sweetest is one of the levulose. The levulose is smallest and that's called levulose. The smallest and the sweetest sugar. Alright. Now this one they can give you the name of that. The basic components are, when I say cellulose, basic components are only glucose. If I say maltose, basic component is only glucose, monomers are. But if I mention lactose or sucrose, they'll give you this pair by pair. If they give you cellulose, they will give you glucose. They won't give you maltose because it's a homopolymer, same molecules attached together. So the monomers of, if they ask you monomers of, I say lactose. The monomers of lactose are, they can give you in pair, glucose, comma, glucose, or three dashes, glucose, glucose, glucose. So there they'll trick you. Why? You should know lactose is a disaccharide, it's not a polysaccharide. So you have to make clear of yourself what is meant by di, what is meant by poly, and what are the things. Mono and hetero, they are all nothing but polymers. If I break the bond between them, it will become a monomer. If I break the bond here, it will become separated glucose and galactose. If I break the bond, it will become glucose and fructose. Do you follow that? So these questions can be direct question asked. Anyhow, then another thing, the 5-carbon sugar, the monomer of a 5-carbon sugar. Example for 5-carbon sugar is the ribose sugar. 4-carbon compound is the glucose, and again 3-carbon compound is the 6-carbon sugar is the glucose. 5-carbon is ribose, deoxyribose. You remove the oxygen from the second carbon. That is the second carbon in the case of ribose. Ribose sugar will be H only. There is no O. That's a deoxyribose sugar. The O will be cut off. That is from the second carbon. And you should know particularly when you say, they can ask you anything if you want to be very tricky. They can ask you the attachment to the 6th carbon, what is attached to the 3rd carbon. Particularly when the ribose sugar comes, it's very, very difficult in a sense. First carbon will be attached something else. Second carbon will be free. Third carbon will be attached to phosphate. Fourth carbon to the fifth. Fifth will be attached to phosphate. The ribose sugar has got in the each corner is pulling out in different direction. Like only one fellow will be free. That is the second carbon. That is where the oxygen is lost. That's why it's called DNA, deoxyribose sugar. The RNA, ribose sugar will be HOH. In DNA, second carbon will be losing the O. This is one of the questions they got. The difference between the molecular structure, the deoxyribose sugar and ribose sugar. Ribose sugar, second carbon is full, HOH. But in the case of deoxy, oxygen. Okay, I'll see you in the next class. Thank you.